The spindle sugar is connected to a base. So this is connected to a base. Spindle sugar is connected to a base. You cannot see that. And this is 1, 2, 3. Phosphoric acid is connected to this 1, 2, 3, 4, 5. That is 5 dash. Phosphoric acid is connected to 5 dash. So I'll be writing of CO. Base H H wedge H wedge CH2OH. Here H. Now phosphorus acid is like this is C dash 1 dash 2 dash 3 dash 4 dash and this is 5 dash. When phosphoric acid is connected to 5 dash position of this sugar, we get a nucleotide. That is the monomer. This is a nucleoside. Base is connected to 1 dash position of the sugar. Then this is called nucleoside, SIDE. When this nucleoside is connected to a phosphoric acid in the sense P, this O, here O, then OH, here it is OH, then it is OH. So this is called OH. This is called a nucleotide. This is called a nucleotide. Nucleotide. This is the monomer. When many monomers are joined together to get a polymer, we get this RNA. Here it is ribose, so RNA. If the O is not there, then DNA. In DNA, bases will be adenine, thymine, cytosine, guanine. This base may be adenine, this base may be thymine, this base may be cytosine, this base may be guanine. So this is 2-deoxyribose. This spindle sugar is 2-deoxy. On top there is no oxygen, deoxy. Bases will be ATCG in the cases of DNA, AUCG in the case of RNA. In that RNA there will be oxygen. That is ribose, ribonucleic acid. In the case of DNA, deoxyribonucleic acid. So this is the monomer. Now next monomer, this will be connected to this monomer will be connected to the 3' position of the next nucleotide. 3' position of the next nucleotide. So there is no space to show. So I will be showing on this 3'. From down, one nucleotide will be connected to the 3' position. Like that, on 3', next one on 3', next one on 3', next one, a polymer will be formed. So I will show like this P, double bond O, OH, and here this is phosphodiester. Up and down, 2 esters. So phosphodiester. This is the CH2 of the next nucleotide down. Or you can say this is connected to the 3' of this next nucleotide. So monomer is nucleotide. Monomer is nucleotide. So here, what we get, this is RNA. RNA, this chain is RNA. If the spore is not there and base, if it is A, T, C or G, then we call it as DNA. Anyway, in RNA and DNA, nucleotides are the monomers. When base is connected to the sugar, then we get nucleoside. When this nucleoside is connected at the 5' position of the sugar with this phosphoric acid, we get nucleotide. This phosphoric acid will be connected to the 3' position of the next 3' position of the sugar of the next nucleotide. Like that, many monomers are joined and we get the polymer. And here, so base, sugar, phosphoric acid. Phosphoric acid. So base is projected. Base, sugar, phosphoric acid. Phosphoric acid. Now next one, base, sugar, phosphate, phosphate. This is connected to the 3' position. So base, sugar, phosphate, base, sugar, phosphate. Next, base, sugar, base, sugar, phosphate. Like that, so many. This is one strand. This is one strand. In the case of RNA, two strands are complementary to each other. Two strands are complementary to each other. So the space, if it is A, it will be pairing with timing of the next strand. So here another strand like this. That is a helical structure. These two strands are going with the right-handed helix, that is alpha helix. So during this pairing, what happens is right-handed helix, two strands will be going like this. So two strands. This is a right-handed helix. So here, the next one will be going like this. This is again a right-handed helix. During this moment, during this, when these two right-handed helices are going like this, pairing of bases are taking place. Pairing of bases. This adenine will be making hydrogen bonds with thymine of the next, or cytosine will be pairing with by hydrogen bonding with guanine. So here, thymine will be pairing with adenine of the next. Guanine will be pairing with cytosine of the next. Like this. Due to this base pairing, suppose there are 40 adenine, there will be 40 thymine. Suppose there are 30 cytosine, there will be 30 guanine. Due to this base pairing principle. In DNA is double stranded. DNA is double stranded. Double stranded. DNA is double stranded. Base pairing. Here there is base pairing. DNA is double stranded. There is base pairing. It gives genetic information. So these are the with adenine, thymine, cytosine, and guanine. Genetic information. These are the characteristics of DNA. It gives genetic information. Now, okay, it has bases adenine, thymine, cytosine, guanine. It is double stranded and base pairing principle is followed. So this is one strand, that strand I have shown like this. And this is another strand, and there will be hydrogen bonding between the bases. Adenine always hydrogen bonds with thymine. Cytosine always hydrogen bonds with guanine. Now, RNA. RNA is single strand. RNA is single strand. In this bases are what? Bases adenine, uracil. If uracil is present on hydrolysis, you'll be getting different units. These 3 units you will be getting on hydrolysis. RNA on hydrolysis, you will be getting sugar. Which sugar? RNA, ribose sugar. You will be getting one phosphoric acid. You will be getting bases. What are bases? Adenine, uracil, thymine, adenine, uracil, cytosine, and guanine. These are the bases obtained when RNA is single stranded. This is protein synthesis. The use of RNA is protein synthesis. Protein synthesis. Then AUCG, adenine, uracil, cytosine, guanine. This is single stranded. Single stranded. Base pairing is not there. Base pairing is not there. So, when RNA is hydrolyzed, there is no relation between the various bases obtained. When RNA is hydrolyzed, no. Here the sugar is what? Ribose. Here the sugar is ribose. Ribose sugar, or in the case of DNA, deoxyribose. So let's see the differences between DNA and RNA. DNA and RNA. Biomolecules. The last biomolecule we are learning is nucleic acid. DNA and RNA. Okay. DNA and RNA. DNA. Sugar is deoxyribose. 2-deoxyribose. 2-deoxyribose. RNA ribose. DNA, genetic information. It gives genetic information. RNA, protein synthesis. The function of RNA, protein synthesis. Protein synthesis. There are three types of RNA. Messenger RNA, transfer RNA, and ribosomal RNA. Different functions. You have to learn only the names. Messenger RNA, that is mRNA, transfer RNA, that is tRNA, and ribosomal RNA. All are having different functions. You have to by heart only the names of these different types of RNA. Okay, genetic information. In DNA, adenine, thymine, cytosine, guanine are the bases. And here, RNA, adenine, uracil, cytosine, guanine are the bases. Now this is double-stranded, and RNA is single-stranded. Now here, base pairing principle is followed. There is no base pairing principle. No base pairing principle. No base pairing principle. So, primary structure and secondary structure. The structure of DNA or RNA that is learned at two stages, primary structure and secondary structure. Primary structure means, again, the specific sequence, the information regarding the sequence of these things. Information regarding the sequence of these nucleotides. Right now, information regarding the sequence of nucleotides is the primary structure. Information regarding the sequence of nucleotides of a nucleic acid is called the primary structure. Information regarding the sequence of nucleotides of a nucleic acid is the primary structure. Now, what is meant by secondary structure? Secondary structure, whether it is single-stranded or double-stranded. Secondary structure, double helix structure. Secondary structure of DNA, that is double helix structure. Secondary structure of RNA is single-stranded. Two strands are complementary to each other because hydrogen bonds are formed between specific pairs of bases. What is the secondary structure of RNA? What is the primary structure of RNA? Information regarding the sequence of various nucleotides of DNA. That is the primary structure. What is the secondary structure? Single-stranded. They are single-stranded. Sometimes they fold back on themselves to form. Means to look like a double helix structure. It is single-stranded. Now, what are the applications of this DNA? The main thing is DNA fingerprinting. Right now, DNA fingerprinting. This is for identification of criminals. Number one, identification of criminals. Number two, to determine the paternity of an individual. To determine the paternity of an individual. To determine the paternity of an individual. That is DNA fingerprinting. By DNA fingerprinting. Number three, to identify dead bodies in accidents. To identify dead bodies in accidents. To identify dead bodies in accidents. Number 4, to identify racial groups. To identify racial groups. To identify racial groups. To identify racial groups. To rewrite biological evolution. To rewrite biological evolution. To rewrite biological evolution. Next question. What products would be formed? What products would be formed when a nucleotide? What products would be formed when a nucleotide, that monomer from DNA, containing thiamine. Thiamine is hydrolyzed. Thiamine is hydrolyzed. Thiamine is hydrolyzed. Number 1, phosphoric acid. Answer, phosphoric acid. 2, if it is DNA, then it will sugar will be beta D 2 deoxyribose. Beta D 2 deoxyribose. On second carbon, no oxygen. Beta means OH is up. Beta D 2 deoxyribose and thiamine. Next question. When RNA is hydrolyzed, there is no relationship among the quantities of, among the quantities of, different bases obtained. What does this fact, suggest about the structure of RNA? What does this fact suggest about the structure of RNA? Answer, RNA is single-stranded. Answer is RNA is single-stranded. Because base pairing principle is not followed. Base pairing principle is not followed here. Base pairing principle is not followed here, unlike DNA. With that, we finish this chapter. Next, we are moving to D block elements. D and F. D and F. D block elements are also called transition elements. The definition of transition elements goes like this, which are having incomplete d orbital, which are just note down. Transition element is defined as, first we are going to D block elements, that is transition. Transition element is defined as atoms with incomplete d orbital. Atoms with incomplete d orbital. It shows the transition from more active alkaline S block metals and less active P block metals. So it is a transition showing from high active to low active, from more active S block elements to less active P block elements. The real definition is like this. Transition element is defined as atoms with incomplete d orbital. That is 1 to 9. D orbital, it can maximum accommodate 10 electrons. Incomplete d orbitals means not 10, 1 to 9 electrons. First, we'll be dealing with D block elements, and then we'll be going to F block elements. D block, first transition series, that is scandium, titanium, vanadium, chromium, manganese, iron, cobalt, nickel, copper, zinc. First transition series, or we can say this as 3D series, where 3D orbitals. 3D orbitals are progressively fit. 3D series. Next, scandium to zinc, yttrium to cadmium, lanthanum to mercury. Yttrium to cadmium, lanthanum to mercury. So, zirconium, hafnium, molybdenum, tungsten, technetium, rhenium, ruthenium, osmium, iridium, palladium, platinum, AgA. So here, first transition series or 3D series, second transition series that is 4D series, third transition series that is 5D series. As we come down, generally we say atomic size increases. In a period, atomic size, when the atomic size is decreasing, the decrease from element to element is little more. Why? Because D electrons are having very poor shape effects. Continue in the next class.